So today I'm going to talk about the development and the evolution of some of nature's strangest woods, which are in lianas. And I integrate classical stem ontogeny techniques with comparative phylogenetics to address the question of the evolution of development of these forms. But before we go to the extremes of nature, let's start from a common place that we're all familiar with, which is the stem development in trees and shrubs. So we're all familiar with this framework, starting from primary growth, going to secondary growth. And so in primary growth, what we see, of course, is discrete vascular bundles in a ring. But if the plant is going to grow in girth and grow laterally, then it needs to produce something called a vascular kingdom. And the vascular kingdom is a combination of two meristematic regions. One from the fascicular cambium, which arises from within the vascular bundle, and the other from the interfascicular cambium that arises from the boundary of stem. I'll use this pointer. When those two cambia from different origins fuse together, they form the vascular cambium that gives rise to whole rings of secondary xylem towards the center of the plant, here in green, and whole rings of secondary phloem towards the outside of the plant. Those respectively are the water-conducting and the sugar-conducting cells of the secondary body. So in looking at this in cross-sectional view, we have here, this is the vascular cambium, and it's sandwiched between the secondary xylem towards the inside and the secondary phloem towards the outside, which is also known as the inner bark. And this is a very extraordinary, complex development, but it's actually just a basic bowel plan that gives rise to the woody growth habits that we see in nature, from tall trees to shrubs to lianas. And we understand that there's a continuity here of secondary growth connecting these three forms. But what actually separates them? What is going on developmentally that separates these three woody growth forms from each other? Specifically, in my case, what separates the climbing habit, the liana habit, from self-supporting plants known as trees and shrubs? In order to think about that more critically, we have to define what lianas actually are. And we understand that lianas do not form a monophyletic group. They're not a lineage, they're not a family. It's just a growth form that's evolved multiple times across the vascular plant phylogeny. So we have it in ferns, we have it in gymnosperms, we have it multiple times in angiosperms. The term wood, using here sensu lato. And with the convergence of this growth form, the convergence of this habit, comes the convergence of an anatomy called the Liana syndrome anatomy, which is three functional components working together in a coordinated fashion to achieve three functions: maximizing conductance, maximizing flexibility, and strength, to our right there. So to maximize conductance, this is something we see very, very common, actually, which is that lianas have larger vessel elements, so they're water-conducting conduits, than trees, and in closely related lineages, and we see this trend time and time again. Together with that, flexibility and strength, I like to think about them working in a coordinated fashion together very tightly, actually. And both of these cases, for Botany and Malpighiales, the blue being non-lignified parenchyma interspersed within the secondary xylem, which is sclerified here in red. And so that's the mixing of soft and hard tissue together to hit the sweet spot of being strong and flexible and also conducting lots of water, because we're a climber. We don't want to bend. We want to bend, we don't want to break. We are all climbers. All right, so we have this idea that the anatomy can be very different than trees and shrubs. But if we push it a little bit further, we push the limit on it, then we can get something called vascular cambial variance, which is an extreme elaboration on this basic bowel plan, giving rise to a very diverse array of stem morphologies and conformations found across different liana lineages. So here we have the irregular stem conformation in Malpighiaceae. We have Celastraceae here showing successive cambia. So those may look like growth rings, but they're not. Each of those arcs here that are darker are a vascular cambium stacked on top of each other, all producing their own secondary xylem phloem. We're going to see this guy again. And we have this really well-characterized type called phloem wedges found in the Bignoniaceae. So if I want to study the evolution of development of this, I want to pick a system that has a comparative framework to it. And that turns out to be already the lineage I was studying. So, and that's Paullinia, which is a Sapindaceae genus. It's a really large genus, 220 species approximately, found in the neotropics with one species in Africa. It has capsular fruits, and you may know really well the seeds, actually, because the seeds are pulverized and used for the production of guarana in Brazil. So that's really great economic importance, actually. But what's really important for this talk is the diversity of stem conformations. As I stated before, it's a good comparative system because we have species that develop regular stem development similar to trees, but we also have this diversity of cambial variants to compare to across a phylogenetic tree. So this leads to my motivating question, which is this. What is the diversity of vascular cambial variants in Paullinia, and how do they develop and evolve? So that's kind of a two-part question, actually, and it employs two different branches of our evolutionary brain to assess this. The first one is dealing with diversity and development, for that, I employ classical stem ontogeny techniques. I use the technique from Barbosa 2010 that for making these sections that are going to be shown for the rest of this presentation. For 3 stages of development, at the primary growth of the shoot apex, the onset of secondary growth when the vascular cambium first arises, and the oldest stems that I can get my hands on, I take 3 sections: the transverse, the tangential, and the radial. And all together that pieces together to figure out the sort of 3D structure that wood actually is. So I'm going to go through each of these stages and some of the characters that have been highlighted in my research. So primary growth at the very top of the plant, the closest to the shoot apex I can get my hands on. We have something very unusual just to begin with, which is that the stem outline is not circular like you would expect in trees and shrubs. Most of the species in Paullinia actually are angular, is what I'm calling it, or stellate, kind of like a star, because they're stars. And why are they stars, though? If we look closer in these lobes, this lobe, this lobe, this lobe, this lobe, this lobe, that's where the concentration of vascular bundles is concentrated. In these arcs, that's where there's an absence of vascular bundles. So there's an unequal distribution of vascular bundles around the circumference of the plant, generating this structure that's unequal in its outline as well. At the onset of secondary growth, so as soon as the cambium is formed, I take note of several characters, also taking note of if a cambial variant is formed, and also if the shape of the outline has changed, in addition to many anatomical characters. In the mature secondary growth, so the oldest stem I could put my hands on, I take a lot of character coding from the International Association of Wood Anatomists checklist, as well as several characters I've had to basically figure out along the way as I saw the variation in the lineage. Just to let you know about my sampling, so I took samples from the field in Panama, as well as I used samples from my colleagues that were stored in ethanol from like the 90s and no one ever cared about them. And I also rehydrated several samples from herbarium vouchers, the stems of those, rehydrated them and treated them the same to get this data. I'm going to highlight 3 anatomical characters I think are pretty awesome. And they all kind of have this dimorphism to them. So the vessels in many Paullinia species take on 2 size classes called vessel dimorphism. We've got these large ones, and we've got these chains of smaller ones. Kind of a backup system if these guys blow out from embolism. So we think. There's fiber dimorphism. What we see here, this thin wall, thick wall, this alternation, the red is lignified fibers, and the blue are septate fibers, which are non-lignified fibers, and they're alternating. And what I really like about this character is I think this is Paullinia's way of reaching that sweet spot of strength and flexibility. The other dimorphism I want to point out is ray dimorphism, which is that there are multiserrate rays that are very wide, as you see in the center here, and then there are uniserrate rays that are just 1 cell wide. But that's anatomy. What about ontogeny? So what I found is there are 6 different ways the stems can develop in Paullinia. We know that it has a sort of tree-like, I'm calling in this chart, tree-like ontogeny that we're all familiar with, perfect cylinder, goes into regular stem development. But we can get that same end point, that same regular stem conformation in the end, starting from an irregular starting point, starting from that angular primary plant body with the unequal distribution of bundles. So those are our regular stem developments. What's really cool is that all of the cambial variants actually all ontogenetically arise from this angular primary plant body. So what that means is there's a connection between irregular primary growth and irregular secondary growth in this lineage. So I'm going to highlight 2 ontogenies to drive this home. The phloem wedge one. All right, this is the one where there's spaces on the circumference that have more secondary phloem than the adjacent region. And this is how it goes. It starts off angular, okay? We zoom in, we see that there's more bundles in these lobes, and then there's less so in this arc. So this is going to be critical. The lobes are going to be the site of the future fascicular cambium. The arcs are the site of the future interfascicular cambium. On the onset of secondary growth, what happens is the site of the interfascicular cambium is going to produce elevated amounts of secondary phloem relative to the activity of the fascicular cambium, meaning that these two cambia that arise from different origins are producing different amounts of cambial products, producing this mass amount of secondary phloem. Starting from another place, compound stems. Starting with an angular primary body as well. We zoom in, we see at the onset of secondary growth, there's a cambium that's forming here and excluding this region. So here's a cambium, it's chugging along, but it excludes this region and three others. In the process of excluding three peripheral regions, each of those peripheral regions will produce their own vascular cambium. Hence why they all have secondary xylem and phloem in each of those cylinders. So in the end state is one stem with 4 cylinders, each with their own cambium. So now what do we know about development? We know that there are 6 different ways to achieve stem development in Paullinia. We know that all of the variant types arise from irregular variant growth in primary growth. So how does this look in evolutionary space? In order to do the second half of the question, we need a phylogenetic framework, meaning we need a phylogeny. So we developed the first phylogeny of Paullinia, and it's arise in the data is 11 different loci, 9 of them being single copy nuclear introns, so highly variable regions. And we took this phylogeny, we took this ontogeny, put them together to estimate the evolution of development. And what we found is that here, I have a couple of things I'm going to share with you. So let's just first get our arrangement together. Primary growth is on the left. Secondary growth is on the right. Primary growth, what we find is that most species are labeled here green, which means that most species are angular actually. Secondary growth on the right, all these different colors, I hope you can keep up. Okay, so let me see if I can help you though. The first thing I want you to know is that the ancestral state of the whole lineage in terms of ontogeny is this variant way to achieve regular growth. Starting with this angular body and then rushing on over to become regular. So it's a little tricky, you don't have to know the plants well. You would think it was just regular altogether. But the other things that we need to know is that each of the cambial variants has actually evolved multiple times, except for the successive cambia. But even these crazy ones, this compound type, we see it 1, 2, 3, 4. Or I think I did that, I don't know how to count. Okay, so at least 4 times. The lobe site, we have it multiple times as well. And it's really interesting that the most developmentally complex is the successive cambia and found in open sandy soils in Brazil, and has only have evolved 1 time in this lineage. In addition to looking at Paullinia, I also wanted to look at the greater Sapindaceae lineage of lianas, because there are 5 other genera of lianas in this family. And I found the same pattern, which is this, that all of the cambial variants arise within lineages that already have irregular primary growth, meaning that the evolution of cambial variants in a lineage is contingent on first evolving irregular primary growth, which was supported by a correlated evolution test of Pago 1994. So what are the evolutionary mechanisms we can use to explain this stuff? And then I have one more slide. I was a moderator this morning. Modularity, I think this stands out as really key actually, which is that we have a complex structure. And if we broke up that complex structure into individual items, we can start to think about how are they working in a coordinated fashion, or how are they actually working independently from each other. And in this case, what I think the 2 modules that are key in explaining the diversity is the interfascicular and the fascicular cambium. Being that in the regular, we're familiar with these 2 cambia doing the same thing in a really tightly and time-dependent and growth rate-dependent way. But the phloem wedges one show us a little step in a different direction, where the interfascicular cambium is producing more secondary phloem. And in the lobe xylem, the fascicular cambium is producing a lot of secondary xylem. So that it's highlighting that these 2 cambia, once again arising from different origins, actually are kind of quasi-independent in a way, And in that way is a mechanism to achieve these different forms of evolution. So I have two conclusions for you that I want you to go home with. In the ontogeny of an individual stem, variant primary growth is the precursor step to all of the cambial variants in Paullinia and within the Sapindaceae. And on the evolutionary perspective, the evolution of these forms is also dependent on first evolving irregular growth. And thank you.